everyone over here is doing absolutely good. And I think slowly, I'm just waiting for sometimes is that everyone comes in. It's sexual reproduction in flowering plants. And you know what? There are almost 13 questions, or basically 13 marks that you can get from this chapter alone in your second PUC examination. And in your case, at almost 3 to 4, sometimes 1 year it went to 7 questions. But recently, if at all we check, it is actually around 3 to 4 questions that you mostly get from this particular chapter. If at all you have to go ahead and dissect it and see that what is exactly inside the flower. Okay, and it's actually very, very, very fascinating because a small little thing undergoes sexual reproduction. It does not even have the brain to do that. Still, it's happening. And don't you think that's nature's, miracle out there? Exactly. So over here, the first part that we need to know, the flower, and if at all we just have a quick zoom in, we will be able to see the male part right over here. These things that you see over here are the male part, and in the middle thing, this particular thing that you see over here is the female reproductive part. Okay, in the male part, you have got the pollen grains. The pollen grains which are there, they are going to come and land on the stigma of the female part. Now, at this point, if you feel that, ma'am, I do not know what is stigma or basically what is happening all around, I can explain you that as well. I think we are good to go with this. Okay, so over here, the female part, it's very simple. Okay, you have something very sticky. You call this particular portion as what? You call this as stigma. Okay, stigma for sticky thing. Now over here you have a long slender something like structure over here which you call as what? Style. And finally at the base you have something else which you call as ovary. And inside the ovary you have one very important thing, that is ovule. And by the end of this session, you are going to remember all these names and what are going to happen very, very easily. For the first time, obviously, for me also, it's very difficult to analyze what you know, what you do not know. Okay, but with time, you'll get to sort things very clearly. Okay, so we have stigma, we have style, and we have the ovary. Inside the ovary, what do we have? We have the ovule. Inside the ovule, what do we have? Inside the ovule, we have the female gamete. Okay, the female gamete. So in human beings also, we have the female and the male gamete. Male gamete is known as sperm, female gamete is known as ovum. Similarly, over here also, there is female gamete, and female gamete is inside the ovule. Okay, that is done. What about the male part, ma'am? So here you spoke about stigma, style, ovary and ovule. Together, what do you call it? First tell me that. So together we can call this particular structure as pistil. And it is also sometimes known as carpel. So if at all you are being asked what are the different parts of pistil, do not be worried. It's the same thing. Stigma, style, ovary. What are the different parts of carpel? It's the same thing. Stigma, style and ovary. That is how it usually goes. Okay. Now we are coming to the male part. And the name of the male part is very easy to remember because in the male part itself, the male name is there. That is stamen. Men stands for men. They are males, definitely right. And the stamen, you just have a long slender like structure like this, and then you have something like this. So this long slender like structure is known as filament. Okay, and this particular part that you see over here is known as anther. So ma'am, you said that here the female gamete is inside the ovule. Similarly, do we have anything over here where the male gametes are going to be there? Obviously, yes. And what is that particular place? It is basically the anther. Inside the anther, you have something that is known as the pollen grain. Okay, you would be having something that is known as the pollen grain. Now, you might think that pollen grain is the gamete. Wrong. Inside the pollen grain, you would be having what? The male gamete. Always understand this. This is a usual confusion that, ma'am, pollen grain itself is a gamete. No, pollen grain has the gamete. Okay, pollen grain has the gamete. And the first part of this particular chapter is where you are going to get inside this whole structure, or basically the structure of this particular thing that is seen over here. So this is the anther and this is the filament in real life. Okay, and over here, this particular portion that you see, this is the stigma and this is going to be the style. Where is the ovary? The ovary will be at the bottom. Okay, within this particular structure, there will be an ovary as well. This is how it usually goes. Until this particular portion, is it clear to all? Until this particular portion, is it clear to all? It will be really good if at all you let me know that. Then it's sorted. Clear? If yes, give me a thumbs up. If no, you can ask me a question as well. So now what we are going to do in this particular chapter is we'll deep dive into this structure and understand things even more better, in a better manner. That is how it is going to be processing. So when I say male part, or basically the other name of it, as you know, it is what? It is stamen. From this point onwards, things are going to get very, very interesting. You have a difficult name for the male part as well. And what do you call this? You call this as androecium. What do you call it? Androecium. Androecium is the male reproductive part. It is also known as the stamen. And as I already told you, it is divided into two parts. One is the filament that you see right over here, and the other one is the anther. Okay, now in your NCERT textbook, something is written which is important for you to know, and you might think that it's not that important, because in NCERT, it does not feel to be that important when I read through it. So listen to me very carefully. This came in a NEET paper actually, okay? And it said that over here, when I say that there is a structure like this, that is the filament, over here, filament has two sides. One end of the filament is known as the proximal end. What do you understand by proximal? Proximal means it is going to be near. Okay, proximal means it is going to be near. Near what? So the flower which is there, if you have seen, it usually have a green structure like this. Right? And what do you call this as? You call this as thalamus. You call this as thalamus. So your filament is attached to the thalamus, and this part of the filament which is attached to the thalamus, what do you call it? You call this as the proximal end of the filament. Right? Now, on the other hand, there is more to it. At the top you see anther. The point of attachment of anther to the filament, you have another term for it. Since it is at distance, you will now call this as what? The distal end. So there is proximal end which is attached to the thalamus, or it can be any other part of the flower which is there. At the distal end, you have one thing for sure, and that is going to be the anther. Please do remember this. This is one very important line of your NCERT, and from this you have got a NEET question as well. Cool. So stamen has two parts, a long slender filament, that is this, a terminal usually bilobed anther. So what do you mean by bilobed and all? I'll come to that. The filament's proximal end is attached to the thalamus, or basically a petal. So sometimes it can be attached to the thalamus like this, or sometimes in some cases, I know it looks very weird, but sometimes over here it can be attached to a petal as well. A petal as well. That's kind of weird, but yes, that also happens. Okay? Until this particular point, is it clear? Is the thing about the filament clear to the one who is watching this? If at all, I see there is a mention about the bilobed anther. Okay, there is a mention about bilobed anther. What do you mean by bilobed anther? So bilobed means, if you see at this particular structure, you will be able to see there is one big line in between, and this particular big line is dividing things into two. So this is one lobe, and this is the next lobe, which is why you will call this as what? A bilobed structure. Okay, this is what? A bilobed structure. Two lobes are there. Now, here also, if at all you zoom in and probably see very, very carefully, you will be able to see one more very important thing. So I'll just draw it first and then I'll show it to you. Okay. So this part was one lobe, this part was one lobe, but in between that there was a line which was coming in between. What is this line? You will call this as the longitudinal groove. Longitudinal means like this, right? In a long manner. Groove means what? There is a gap in between. That is what you mean by groove. So basically, it actually looks like this. That is, you have a lobe, okay? You would be having a lobe, and in between the lobe is also divided into two. The lobe is also going to be divided into two. So in that particular case, if I say that this is lobe 1, and basically this is lobe 2. In lobe 2, there is another division, okay, another division, one share. One right over here is known as what? First part, and the second part is over here. Do you have a name for that, ma'am? Obviously, yes, we do have a name for that. What do you call this? You will call this as theca. What do you call this? You will call this as theca. So one lobe has two theca. Another lobe also has two theca, which is why we say it is dithecous in nature. What do we say? We say it is dithecous in nature. So initially we learned that, ma'am, we know that basically the anther over here is a bilobed structure. And why did ma'am say that it's a bilobed structure? Because here I can clearly see this is one lobe and basically this is going to be the second lobe. That is why I said it's bilobed. Now I'm saying that basically it is also dithecous in nature. Now why do you think I will call it dithecous in nature? Because over here, my dear children, if at all you see this one lobe over here, there is difference here. There is basically a gap being maintained over here, dividing the lobe also into two. So one lobe into two thecas, which is why we will call this as dithecous in condition. Okay, we will call it as dithecous in condition. And this particular thing that basically separates the two theca, I'll say it is a longitudinal groove running lengthwise separating the theca. It is running lengthwise, not widthwise, running lengthwise separating both the theca out there. Got it? And finally, the anther is a four-sided tetragonal structure consisting of four microsporangia. My God, that is way too much for us to study, right? So how do we study that? It's a very easy way to study that, and I'll tell you. So over here, you have to imagine, okay? Use your imagination, right? So what if I cut it like this? What if Jasmine Ma'am goes and cut it like this? What is going to happen if I do that? Nothing, ma'am. The plant is going to, or basically this particular thing is not going to function anymore. That's the only thing. That's fine. But what if I look at it from the top? So the top view, basically, it is going to look something like this. It is going to look something like this. So if at all you have got an understanding, this is going to be one lobe, this is the another lobe, and as I told you, one lobe has two theca. So this is one theca, this is another theca, this is another theca, this is another theca. So dithecous here also, right? And this is the thing that is mentioned over here. The anther is four-sided. Four-sided, this is the first side, second side, third side, fourth side, which is why I'll call this as what? A tetragonal structure. Tetra means four. Okay, tetra means basically four. So it's a four-sided structure which is there, and it consists of four microsporangia located at the corners, two in each lobe. Now, what is that? What is microsporangia over here? So microsporangia is basically the place where the pollen grains are going to be formed. I told you inside the anther you have the pollen grain. Inside the pollen grain, what do you have? You have the gametes. So you need a place for the pollen grains to develop, right? And that place is basically this microsporangia. So I am right now telling you that when you talk about each corner has basically one microsporangia. So how many total microsporangia are there? If each corner has one microsporangia, so in total, how many microsporangia is going to be there? 1, 2, 3, 4. We have 4 microsporangia as well. Very, very, very important. So the anther is four-sided, tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. In each lobe 2, in each lobe 2. Right? The microsporangia develop further and becomes pollen sacs. So this particular microsporangia which is there, later on, as I said, that this is where the pollen grains are going to be formed. So which is why it will later on transform into what? Pollen sacs. This is where the pollen grains are going to be formed. So Nisagra or anyone who is probably listening to the session, are you able to understand this? We will be having few layers, and these layers are very important for you to remember because you mostly get so many questions from this particular topic. It goes like this. You all might have went to a ENT doctor, ear, nose, throat doctor, right? So similarly, you have to remember like this. There is EMT. Instead of NT, it is going to be what? MT. EMT is the code that you need to remember. I told you that in each of the corners you have microsporangia. So basically let me make something like this right over here. Okay, done. Now I'm going to talk about the wall layers, and it goes like this. So there is the outermost wall layer out there which starts with E, and can you guess the name of it? Anyone probably watching this video, can you guess the name of it? What do you call this particular layer as? What do you call this particular layer as? It's very simple. Any idea? The outermost layer of the anther over here? Epidermis, exactly, perfect. It is known as epidermis. Cool. Now, going ahead, inside that, what do we have? Let's see. So, inside that, there is another layer which again starts with E because that is a logic. And that particular layer, any idea? What do you call it? Any idea? If not, I can tell. Definitely. But I want you guys to try. Or probably at least look into your NCRT. The next layer, endo. It is known as endothecium. Okay, it is known as endothecium. Cool? Yes, perfect, Nisarga. Okay, now over here we have a layer, but I'm going to skip that layer for some time right now. And I'm going to talk about something else altogether. This particular area that you see is actually filled with lot of cells. It is actually filled with lot of cells and is super important. And since it is super important, it starts with S. It starts with S. First of all, what do you call this as? You call this as sporogenous tissue. You will be calling this as sporogenous tissue, and the reason of calling it this tissue is because it is made up of so many cells. Now, what cells? These cells are known as pollen mother cell. Or I can also call this as PMC. So, ma'am, you were saying that in the anther, the sperms are, sorry, not the sperms, the pollen grains are going to be formed, and this is what you mean. So, basically, in the anther, there is mother cells for pollen grain, and these mother cells probably might be giving rise to the pollen grains later on. If that's your thought, the answer is yes. These pollen mother cells are the ones which are going to divide further and are going to give rise to the pollen grains later on. And you have one more name for it. Okay, it is also known as pollen mother cell. Apart from that, it is also known as microspore mother cell. It is also known as microspore mother cell. So, in the microsporangium, you have the microspores out there. Okay. So, in that case, it will be also known as MMC. But, ma'am, you said that they are going to give rise to pollen grains. But how can you call them as microspore mother cell? Are they forming something like this known as microspore as well? The answer is yes. So, basically, first of all, when these guys are going to divide, they will form something that is known as microspore. Okay, they will form something that is known as microspore. This microspore later is going to transform into what? Your pollen grains. So in that case, saying it as microspore mother cell is not wrong. Calling it as pollen mother cell is also not wrong. Why? Because anyway, it is going to make pollen one day, and before that, it is making microspore as well. Okay, so this is the sporogenous tissue which is there, and this is very important. Now, this very super important guy has to be saved, right? It has to be made sure that everything is super cool, that these cells get the nourishment that they require such that it can form the pollen grains later on, which is exactly why you have an important layer surrounding this particular thing. And what do you call this layer as? Any idea what do you call this layer as? So this layer has basically cells which are like this, and these cells, mostly if at all you see, they have so much amount of tapetum. Exactly, it is known as the tapetum. So the T over there, we are decoding it, tapetum. And the important thing about tapetum is that tapetum is there for nourishing the pollen mother cells of the microspore mother cells. Okay, so they are for their important function is what? Nourishment. That is one thing that we got to know. Apart from nourishment, what is the next thing that we need to remember about them? They have so much cytoplasm. Cytoplasm, why? Because cytoplasm, if at all it is dense, that means it can provide enough amount of nutrition. That is why they have what? Dense cytoplasm. Dense cytoplasm. Now, to have dense cytoplasm, you also think, okay, if one cell is there, one cell with one nucleus will be having a little less cytoplasm, a normal cell, right? Now, if at all I want to make sure that my cytoplasm is more, probably I'll think of something. That is, I was one cell, but I made others a fool by saying that I am going to divide. My nucleus divided, my cytoplasm also increased in amount, but then cell division did not take place. Cell division did not take place. I took all of it for myself. The only problem that I have right now is basically I will be having two nucleus. I will be binucleate in condition. Binucleate means basically you will be having two nucleus. Why? Because actually this cell should have been divided into two, but it didn't. Okay, the cell which was there, it increased the cytoplasm, saying that, okay, I have divided my nucleus, I will divide the cell also. But they did not let this particular division happen. So karyokinesis took place, as in basically the nuclear division. The nuclear division or karyokinesis, that has taken place, but the cytoplasmic division didn't take place. Cytokinesis didn't take place. So I'll write it down. No, I was writing with rubber. Oh my God. No cytokinesis. No cytokinesis. Kinesis means division. Okay, that is why they are having two nucleus. These features that I am talking about right now, they are super important features of what? Of this particular guy, that is the tapetum. Okay, until now all clear? Now, okay, I'll ask you once more after I talk about this middle guy in between. Okay, then I'll ask you, is it clear or not? So now we have one more layer. So we have sorted about the E, that is epidermis, E, endothelium, T, tapetum. Now what about M? So M guy is basically in middle. It is right in the middle, and what do you call this as? You will call this as the middle layer. Okay, you are going to call this as what? The middle layer, and middle layer is also important. Now, if at all you ask me, ma'am, actually, what is the function of these guys? Okay, tapetum I got is there for nourishment. But what about epidermis, endothelium and middle layer? So you only imagine, okay, if at all you want to protect this very important sporogenous tissue which is having so many cells, don't you think we need protective layers? Yes, definitely we will need it in protective layers. So basically, these three layers which are there, listen to me very carefully, this is important. Okay, these three layers which are there right over here, they are for protection. And they are also for one very important thing, and that is known as dehiscence. What do you mean by dehiscence? Basically, breakdown of anther. If at all they are not supportive, basically you cannot break down the anther. But why do you want to break down the anther? That's so unfair. It's because the pollen grains are going to be made up over here. The pollen grains have to leave here to go and land on some stigma of a female reproductive part, right? Which is exactly why you need dehiscence. And for dehiscence to happen, basically you will need help of these three layers, because these three layers are the ones that are going to break down. So I'll say they are there for protection, as well as the second function of these guys is basically for dehiscence. It is also there for dehiscence. Always remember that. Now what about tapetum? Tapetum, so we studied, it is there for what? Tapetum, we studied, it is there for nourishment. So if you're asked which layers are there for protection and dehiscence, only three layers. Which layer is there for nourishment? It will be tapetum as well. Is this thing clear to you? So the question goes like this. Consider the following statement and choose the correct answer from the given option. Statement number 1. Innermost layer of microsporangium is tapetum. Statement number 2. Cells of tapetum possess dense cytoplasm, more than one nucleus, and nourishes the developing pollen grains. What do you think is the answer? Okay, within 17 seconds, someone gave me the answer. Perfect. 17 seconds. Nisarga, perfect. Actually, your name is Nisarga, or is it something like the S stands for something? That's your surname? So I should not have given 60 seconds, right? I gave because in case that you get 60 seconds, technically speaking, you get 60 seconds for a question. That is the exact reason but I gave you. Probably the next question that I am going to show you are not going to require that much time. So just a second, I'll end the quiz. Okay. So let's see the result. Perfect. Now, going ahead with the next thing which is up there. So the answer for this question is going to be B. The reason for that is innermost layer of microsporangium is actually tapetum, EEMT, last one. The cells of the tapetum possess dense cytoplasm, more than one nucleus, also binucleate, and nourishes the developing pollen grains. And this is 2021 question, a very recent one. So probably this gives a better understanding. Now, this is the next question that is there, and you have to think, okay? Think for this question and then only give me the answer for this. Now, for this, again, I am going to probably... Okay, this time I'm going to give you less time. Let's give 30 seconds. And the question goes like this. Let's go. How How many microsporangia are located at the corners of a typical bilobed anther of angiosperm? Pollen mother cell happens, produces microspores. Now microspore tetrad which is there initially is grouped in 4, but later on they separate. And when they separate, basically anther dehydrates, microspores dissociate and develop into pollen grains. And so many pollen grains are going to be formed up right over here. Now, I have a question for you, and again, this is something. Okay, so yes, I think we can solve it up. Let's go. Quick question. In microsporogenesis, the pollen mother cell, okay, form microspore through mitosis, form microspore through meiosis, undergo mitosis and forms 2 similar size cells, undergo meiosis and form 2 similar size cells. What do you think is the answer for this? And the timer starts right now. Let's see. It's an easy question, right? This also was a KCET question. Okay, the answer is correct, and probably I can just end up the quiz. Cool, perfect. Just give me a second. Yes. Fine. So the answer for this question, we all know, is to form microspore through meiosis. Okay, it is to form microspore through meiosis. We just studied it. There are 2 rounds of meiosis that happens right over here. Meiosis 1, meiosis 2. There is no mitosis that is happening until this particular point which is there. So the answer is B. I think there is one more question. And for this question, I think you will require time. Okay, you might require time, which is why I'll probably just increase the time slot. Just give me a second. Us has to get over. Let's see. Okay, so pollen as an allergen and a performance booster, what do you mean by that? So many people are kind of really allergic to pollen grains. Okay, and that means if at all they see something like pollen grains, they are very small particles, right? They can move around. They're just 25 to 50 micrometers. Sometimes we go like chee, like that, right? So basically pollen allergies are very common, and it can cause very different varieties of problems. That is either asthma or probably bronchitis or any other respiratory disorders which are there. Ma'am, do we have to remember it? No, just understand it. Okay, you will understand it. You do not have to mug up things. Asthma, bronchitis, basically it's everything related to your respiratory system. If at all you're taking in this particular thing, that is pollen grains, you might be allergic to it and you might respond to it in that way. Cool. Now, next is, okay, the one that causes severe pollen allergies. So this particular guy, which is there, I'm pretty sure that you have seen it. Okay, in the roadside and all, you might have seen the small, where is it? The small flower-shaped structure like this. White color flower would be there. The previous year I showed it. If at all I can, in tomorrow's class, probably I'll bring that flower, if I can. Okay, so this is known as Parthenium, and it's basically also known as carrot grass. Parthenium or carrot grass, this is one of the invasive weed. Invasive means it was not from India. Someone somewhere accidentally got it from outside. Okay, or probably accidentally or not, I do not know, but they got it from outside. This is not our country's thing. It came as a wild thing which is over here now, causing severe allergies in our country, that is India. But do know that apart from being the bad part, apart from having the bad part in the story, pollen grains are also sometimes very good. Good in the sense what? Since they are having so much nutrition reserved for, the generative cell out there, they're very nutritious. So pollen grains are highly nutritious and used as food supplements as well. Okay, pollen products are there such as tablets, syrups, which are very popular in Western countries. Now you will not believe me if at all I tell you that you had got a NEET question, even though this is not a NEET class, but I'm just telling you, I saw this question. It was popular in which place? Western countries, Eastern countries, Southern countries, Western countries. Okay, East or West, pollen grain is the best. Okay, West and best. You can remember like that. Okay, so Western countries is where you have the pollen products such as this and this, tablets and serums. And sometimes athletes who are there, they, for increasing their performance, basically this is also acting as a performance booster in the case of athletes as well as horse riders. So this is a small story and I hope you have got it. I don't think there's much explanation needed to it. Okay, but remember it. Now, going ahead with something that is important, and with that we are going to complete the male part. Okay, we are going to complete the male part, and it goes like this. Is it clear, the pollen allergy part? Is it clear? If it's clear, you do not have any doubt, can you say all cool, ma'am? if you have any doubt, please feel free to ask me, okay? Before losing viability, so pollen must land on the stigma before losing its superpower or forming the pollen tube and basically transferring the male gametes within. So basically, I mentioned viability is what? Having the capacity to live, to grow, to germinate, as in to form the pollen tube out there and do the process of fertilization. That is known as viability. So basically, here they have given examples of plants which loses viability like this, and they have given examples of plants which stays viable for some time. Okay, let's see which has the least viability. The things that we usually eat, rice and wheat. Okay, rice and wheat, these plants which are there, they are having very short-lived viability. So short-lived viability as in how much short? Only 30 minutes. They are going to be viable only for 30 minutes. Rice and wheat. So you can just remember like this. After having food, within next 30 minutes, again you feel hungry. So that is how you can remember this most probably. Because you are going to get confused on the day of examination because you have much more examples into that. Right? There's examples of plants which have long-lived viability. Now how do you remember which one had short and which one had long? So remember rice and wheat, you had it, rice and chapati, you had it, but within 30 minutes, mama, I'm feeling again, I'm very hungry. Like that you can remember. Okay, now long-lived viability is basically 3 type of plant family, and I've given the pictures over there, okay, over here. So one is going to be Rosaceae. Okay, one is Rosaceae. The second one is Leguminosae. That means leguminous plants, pea plant, beans and stuff like that. Leguminosae. And the last one is Solanaceae. Solanaceae is basically where you have the potato or basically you have chili. All of it, they come under Solanaceae. So what do we have? We have R, L, S. If you have a way to remember it, yes, please let me know as well. Okay, I will use that somewhere else probably. It is RLS. Do remember they have the highest, or basically they have long-lived viability. They can remember viable for months. They can be remaining viable for months out there. Cool. Now, ma'am, I have a question. Basically, there is one plant which is it's probably at the verge of getting extinct. I want to save this plant and it's a really good plant. What do? I can do one thing, that is I can do cryopreservation. Cryopreservation means you are going to store things in liquid nitrogen. Okay, and the pollen grains can be stored in liquid nitrogen. Why liquid nitrogen? Basically, this is somewhere liquid nitrogen, as usual, it maintains minus 196 degrees Celsius. Okay, it maintains minus 196 degrees Celsius. So when I say liquid nitrogen, so you should understand that it will be in minus 196 degrees Celsius. And in this much temperature, it's too cold, right? In this much temperature, the pollen grains, whatever you try to store, they remain like that. Okay, they are not dying. All the metabolic activities which are going to happen, and there isn't basically all the activities that is going to happen within the cell, it is going to stop. They are not going to die, and it can be preserved like that. So there is a way of preserving pollen grains as well, and that is with the help of cryopreservation, with using what? Liquid nitrogen at what degree Celsius? Minus 196 degree Celsius. Okay, liquid nitrogen maintains that. It's not like you have to maintain the temperature. If you're saying liquid nitrogen, it will maintain minus 196 degree Celsius. It's how it works. Okay, cool. Now, pollen banks are also there, where basically pollen banks means you give the pollen and then you get money. Now, pollen banks is a place where basically the pollen grains are being stored. Now, that can be used for different programs, crop improvement programs later on. I want that crop, I want this crop, I want to fuse them, I want to do this. All of it can be done very easily with the help of this. Got it? Is it clear? Hiyo. Is this part clear? Do I need to repeat it? If it's clear, can you give me a thumbs up? If you have a doubt, can you ask me? Or if it's clear, then we'll move on to the next topic. But remember, with this, we are ending the male part. There is nothing about the male part which is left behind. Doubt? No doubt? All clear? Not clear? Waiting. You have cells arranged like this. What are you going to call these cells as? Basically, you call these cells as cells of nucellus. You are going to call this the cells of nucellus. Basically, one among these cells, okay, one cell among these cells is going to later on going to form what? The megaspore right there. Until now, we didn't see where is megaspore. You just call this megasporangium, but you didn't mention megaspore, right? So basically, this cell right over here, which is a part of the nucleus, this cell, only one cell is there. In the male part, you saw so many cells were there in the sporangious tissue, but here only one cell is there. And what do you call it? You still call it MMC, but this time here the M stands for megaspore mother cell. You will call the MMC, M stands for megaspore mother cell. Apart from, you have the nucleus. Why? To provide nourishment for this. To provide nourishment for this, to make sure that everything happens smoothly over here. That it later divides to form the megaspore, and the megaspore later forms the gametes and all of it. Got it? Is it clear, children? You have to let me know, otherwise I'll not understand that you are getting it or not. Clear? Not clear? Yes, ma'am. Perfect. Thank you, Machli. Got it. So now we come back to this. So nucleus is what? Mass of cells with reserve food, and this food is basically for making sure the nourishment is happening. Cool. Now, before we go ahead with probably the process of megasporogenesis, as in how everything divides, I don't think that we can complete it right in this particular video. Probably tomorrow we'll do that, but we'll still complete the initial part of the female system. Okay, so we saw that the female part is known as carpel or pistil, right? So there are conditions where you have a single pistil. Only a single pistil is there. This is known as monocarpellary condition. Single pistil means basically you just have one of this, only one. This is known as monocarpellary condition. Now you have something else that is known as multicarpellary condition. What do you mean by that? There are so many carpels, not one, there are going to be many of the carpels. And when that happens, there are two things that can also happen along with it. These carpels which are there, just imagine, use your imagination, okay? They have the stigma and the style. The stigma, the top portion, and the style. It's all separate, but they have only one ovule. One ovary. All of it has basically different stigma and the style, but they are fused together in the ovary part. This condition is known as syncarpous condition. What do you call it? Syncarpous condition. Okay, and where do you see it? You see it over here in, what do you say, opium. You might have heard about opium poppy. You will study in this particular grade itself. 12th grade, you have human health and disease, where you're going to see about the drugs. Okay, so from opium you get heroin and drugs like that. So this is the plant that we have, okay, of the opium, and in this they have mentioned about the syncarpous ovary and basically this. So here you have so many stigma in the style. Okay, there is so many stigma and the style. But within it, inside it, you are going to have a single, this is the part, okay, this is stigma, this is basically the male part of the plant. This is the female part, the stigma and the style will be there, but basically within it, they will be having a fused ovary. This condition is known as syncarpous, and basically this particular plant has a name. Opium poppy is a random name that we have, but the scientific name of it is papaware. You know, Tupperware bottle is there, no? Similarly, you can remember papaware. Okay, so papaware. So basically a kid is asking, where is my papa? Okay, like that you can think. Papaware, syncarpous ovary will be there. Now another condition is basically apocarpous condition. Apocarpous condition is what? Baba, over here there will be so many stigma style and ovaries are also going to be separate. They are not fused, they are going to be separate. Where do you see it? In your NCERT, again, this picture is given. Now, you tell me, it's such a boring picture that they have given in the NCERT, okay? In actual, it looks like this, but still it is boring. But do you know, it's of this particular flower. You might have seen it in roadside on some, or probably in some houses. It smells so good. It's known as champak, okay? In Hindi, it is known as champak. In Malayalam, it is known as chembagam. I'm a Malayali, so I can talk about that. So chembagam, it smells so good. So basically within this particular flower, you have this thing where the stigma and the style, everything is separate. They are not together, they are going to be separate. Okay, so that is apocarpous condition, where basically free pistils are there, and you see this in Michelia. This is the plant's actual name. The scientific name is going to be Michelia. So you can remember Michelle, basically has everything for her own. Okay, Michelle has everything for her own. Michelle is someone's name. You can remember that. So Michelia is apocarpus. They have everything of their own. Ovary, stigma, everything is separate. But Papa Bear is having what's in carpus? Ovary. Is this clear? Again, questions have come. Okay, again, questions have come. That is why I'm explaining you this. Fine? Clear? If yes, can you give me a thumbs up? You. Yes. If this part of the story is also clear, just give me a thumbs up. I'll understand that. Okay. Bacha log are understanding. Otherwise, if you do not say, as I said, yes, clear. Perfect. Thank you, Machli. Nisarg, Ga, where are you? Okay. Let's go. Now, one more thing. Small thing is left behind, and that is this. So, in some condition, no, inside the ovary. Now, you have to think inside the ovary. Okay. Inside the ovary, you basically can have either one ovule or can have multiple ovule. Okay, I'll tell you one more important thing before I go ahead. So this is the ovary, this is the ovule. You know what happens? Basically, suppose the male and the female gamete is here, the male gamete comes and fuses and fertilization happens. The ovule which is there, it later transforms into a seed, or basically it forms a seed. What about the ovary? The ovary is going to become the fruit. The ovary becomes the fruit, and Baba, the ovule which is there, it is going to form the seed. That is how things are going to go. Okay? Now, over here, if at all I say that there are multiple seeds, for example, if you think about okay, I hope you have not seen the examples. Okay, basically, if you think about watermelon or papaya, you see so many seeds out there. What do you mean by that? That they were all ovules. So there are chances multiple ovules could be there. Example is in papaya, watermelon. The thing that is difficult for you to remember would be orchid, because that was, you might have not seen the orchid fruit, right? So basically, orchid is something that you might have to remember. Please do remember that. So how do I remember it? PW. Oh, okay. So physics wala, oh, you can remember that. No problem. There's a friendly rivalry that is ongoing. Okay, we do not have any enmity. So yes, PW. Oh, you can remember like that. Remember it, because again, this was also a question. Orchid. Now there are chances that there can be a single ovule as well, and that is this. It is found in wheat, paddy, mango, and things like that. Wherever you find only one seed. In mango, you know, there is only one seed, right? You have eaten mango. So that is, single ovule. Apart from that, wheat and paddy also basically is just single ovule only. That is why we get the single thing. Got it? So please do remember that as well. That was the only thing that I wanted to tell you. And I think with that, we can probably go ahead and solve some questions. Yes, we can solve at least a few questions, and then we can end this particular class. And then in the next session, I'll come back and I'll explain you what is megasporogenesis, and we will also get to understand what is this thing. What is what? Megasporogenesis? Or do you want me to explain it now? I can. I don't mind extending my class. But if you want me to end it up right over here, I can do that as well. In that case, we can solve probably the questions later on, and I can explain you this. Either way, whatever you say, I'll obey that. What do you say? Next class, that is tomorrow, or today only we'll study megasporogenesis. Next class, ma'am. Okay, ma'am, I'm super tired. There's too much in my brain. I cannot take it. I do get it. Okay, so even my energy level has gone a little low. So next class. Okay, perfect, guys, as you guys say. Okay, cool. Let's solve this question then. I hope we're ready with the question. And let me just look through it, okay? Yeah, I can just see the answer like the... Okay, fine. Let's see. I'll give you a time of probably... Okay, before I teach you this, or probably go ahead with this. By the way, will you be able to solve this question by yourself, or because I think I need to show you some picture? If you can solve it by yourself, it is going to be all cool. Let's try. Okay, you try it for some time, and then probably I'll come back. So just a second, let me see first. Okay. I'll give you 45 seconds for this. Let's go. Start quiz. 8 seconds. Oh my God. 3 seconds. Sorry, 2 seconds and 3 seconds you needed to give me the answer. My God, you guys are brilliant. Okay. You guys are brilliant in that case. Fine. I was thinking I'll show you the picture. Okay. So there's an easy way for getting this answer correct. So listen to me very carefully. First of all, yeah. So in this particular picture, as I told you, this is an anatropous ovule, right? And they had clearly put that over here. In the anatropous ovule, this part that you see is known as the funicle. Okay, and the funicle attached over here, and this part where the attachment happens, it is going to be known as the hilum. But if you feel very confusing, basically we'll go with the C1. C, we all know if this is micropyle, this is the opening that we have, which is there. C is definitely going to be the chalaza. Okay, C is going to be chalaza. So basically, if at all we see, it is okay. C option. Okay, C or okay. Just this thing. So over here, C is chalaza, C is chalaza, C is chalaza. I'll exclude this option. This is going to be odd one out. This is going to be the odd one out. Let me see. Okay, just a second. Machli. So yeah. Now we go ahead with B and D and A. Which is the easiest for us to find? Basically A. That is the funicle. Again over here, A is given as nucleus. Nucleus cannot be here, so this option is also going to go. Okay, now we are left with these two options. Funicle and funicle right over here. Now we have nucleus, and after that we have basically, we have nucleus right over here for B option. B anyway has to be nucleus in that particular case. If they have mentioned both the options as nucleus, it will be nucleus only. That means the outer layer of cells which is there. Cool. Now we have one more thing, that is embryo sac and micropyle. So D option which is given. Micropyle is where, Baba? Micropyle is going to be here. Okay. Embryo sac you have not studied until now. But do understand, embryo sac is a structure which is formed inside. Basically, the megaspore mother cell divides, later form megaspore, later forms embryo sac. That is how it happens. And inside the embryo sac, you have the female gamete. So D, which is there, it is going to be embryo sac, which is exactly what I was thinking that should I be giving answer. Anyways, so the answer for this particular question, I think, did I mark it as D? Sorry. The answer for this question is C. It is going to be C. Okay. It is going to be C. Why? Because this is the embryo sac and cannot be the micropyle. I'm so sorry. Cool. How many pollen grains would be produced by the flower? This is the question. If you want, you can take a screenshot of this, or anyway, you'll get the PDF. So just solve it. Don't tell the answer now. Okay? Fine. Was the class fine? Did you understand everything? Did you like the class? Do let me know right now, and then we'll wind it up. All cool? Any doubts? Machli and Nisarga, any doubts that you children are having? I think I'm getting no ma'am. Okay. No ma'am, you didn't like the class, or basically no doubts? I'm just kidding. It's okay. So no doubts is what I understood. Okay then, understood the class easily ma'am. Thank you so much Nisarga. Fine then, you guys have a very good day, and I'm hoping that you will be there for the next class as well. And lots of love to all of you, and take care. Have a good day. And there's the next class. Take care of that as well. Bye-bye.